Good afternoon. I'm very glad to be included today in our great conference. Dear colleagues, dear ladies and gentlemen, dear collaborators, добрый день, здравствуйте. I'm very glad to get this opportunity, this great privilege to deal our results with your respected participants. So thank you, organizers, to give me this opportunity to share my results of my research group with all of you. I will talk today about some bile acid derivatives and salts in drug development and drug delivery. Some things will be general, but some things will be results of my research groups and my collaborators. Bile acids are synthesized from cholesterol in two ways, in two synthetic strategies. First is more common for humans. This is a neutral pathway, and in that pathway, cholesterol is 7-alpha hydroxylated in first step with CYP7A1 enzyme. Another alternative pathway is acidic pathway, where the first step is oxidizing of cholesterol into 27-hydroxycholesterol, and then that compound, that molecule is 7-alpha hydroxylated by a CYP7B1 enzyme. At the end of both these pathways, synthetic pathways, primary bile acids are synthesized, namely henodeoxycholic and cholic acid. In the intestine, these primary bile acids are then converted into secondary bile acids, namely lipocholic and deoxycholic acids, via 7-alpha dehydroxylation. Above these synthetic pathways, cholesterol is starting compound for synthesis of all other steroids in the organism, namely corticosteroids and sex hormones. Bile acids have many different activities in the organism. First of all, and the most known about that is lipid solubilization during digestion through formation of mixed micelles. Bile acids also are included in lipid and glucose homeostasis through activation of farnesyl X receptor and membrane receptors TGR5. Then bile acids is included in bile acid synthesis and metabolism in plasma, in regulation actually. Then bile acids are included in apoptosis, inflammation, and carcinogenesis processes through activation of signaling pathways. Above that, bile acids are included in anti-inflammatory effects through glucocorticoid receptors modifying and treatment of different diseases and states. So all of these activities of bile acids pointed bile acids as lead structures. So many bile acid derivatives were synthesized during many years and decades. And above these bile acid derivatives, there are many very biologically and pharmacologically active bile acid derivatives. Some of them affecting FXR and TGR5 receptors, some of them affecting glucocorticoid receptors or carbonic anhydrase. Some of these bile acid derivatives are used for treatment of metabolic disorders. Some are antibacterial or antifungal agents. Some are anti-cancer agents. So many of them are used today, and many of them are in some phase of preclinical or clinical studies. Drug development could be roughly divided into chemistry-driven and biology-driven, let's say, strategies. In chemistry-driven and biology-driven strategies, order is a little bit reversed in these two strategies, at least at first steps. Namely, in chemistry-driven drug development, first compounds are synthesized according to the similarity to hormones or substrates, or known and used anti-hormones or enzyme inhibitors. After that, next step is testing of biological or pharmacological effect, but for that particular activity which is this compound is synthesized for. After that, other tests can be performed. And in biology-driven drug development, first biological or pharmacological test is performed for many compounds. Then when recognized are leading compounds or lead compounds, it is next step is to test to find a mechanism of action of this compound for one side, and for another side to synthesize some isosteres or bioisosteres compound of this leading compound and to test their activity, so to find even better, even more effective compounds. So computing could be very useful in both strategies, and today is widely used. So in both strategies, many drugs are found and used today. In chemistry-driven bile acid-based drug development, some of effects, some known effects of bile acids are used as a base. So derivatives which are synthesized next are could have agonistic or antagonistic effect of some of these in some tests. So there are many different examples. So I selected only a couple of them. Henodeoxycholic is binding to farnesoid X receptor and pronounce some metabolic effects. On the other hand, 5-alpha derivatives of henodeoxycholic acid with hydroxyl function in side chain. These compounds, such compounds, are antagonists of this receptor. So not agonist, but antagonist. It is probably because of this 5-alpha. 5-alpha configuration. That next, ursodeoxycholic acid is known as an agent for the treatment of glucocorticoid receptor-mediated hepatic inflammatory diseases. On the other hand, its amides are known also as modulators of this receptor. There are many different amides. I selected only one as an example, but usually derivatives are less toxic, less membranotoxic or cytotoxic compared to original bile acids. Next, bile acids are known to bind to glucocorticoid receptors, and by modulating this receptor, these compounds are these androgen compounds are included in the inflammatory processes. So if they are changing, their structures are changing, so they could give some derivatives with less toxicity and with the same effect or antagonistic effect. It is very important, and many such compounds are used. So this is holanic acid. synthesized by Wolff-Gishner reduction synthesis in microwave-assisted synthesis from bile acids via free oxoderivatives of these bile acids. So this compound is also binding to glucocorticoid receptor, and many amides are also binding to glucocorticoid receptor. So this is not this is very important that many compounds can bind to some receptors, but effect in the organism should be tested further. Next, many compounds are derivatives of bile acids, but their structural modifications are very small but very effective in reducing toxicity. I told before that bile acids are membrane toxic. So, for example, such compound is in clinical trials, and it has only one methyl group and one ethyl group in position 6, more than holic acid. But it has similar detergency like holic acid because all 3 hydroxy functions state, but it has a better metabolic stability caused by alkyl moieties inhibiting 7-alpha dehydroxylation in the organism. And this compound is in clinical studies, in clinical trials, because it can increase energy expenditure. So it could be even some kind of supplement or a drug in treating obesity. In biology-driven drug development, always are present some syntheses which are usually not very time-consuming or too complicated, because biology-driven drug development is based on usually is based on the testing of huge libraries of biomolecules, or molecules which could be effective for something. But many compounds are synthesized today with click chemistry. Many of them has this triazole scaffold, 1, 2, 3 triazole scaffold, because it is not very demanding. Usually, is not very demanding for the synthesis, but it is not recognized by the enzyme in the organism. So some compounds which are connected via this scaffold can live longer in the organism, so they can perform their activities. Usually 2 active compounds are connected in that way. So in this bile acid drug development, we have some bile acid or bile acid derivative and another pharmacophore unit. So there are many such compounds connected in that way. On the other hand, condensation reactions are always used for the synthesis of potentially active bile acid derivatives. So I told before that many amides are synthesized, many esters, many amines, or similar simply synthesized compounds. So if bile acid is connected with another pharmacophore, this compound could be even more effective than original one compounds. So, for example, conjugates of bile acids with cytarabine, which is deoxycytidine analog, used for treatment of some types of blood cells cancer. This compound is effective even more than this conjugate is effective even more than alone compounds, and more stable and more bioavailable. More stable then means more bioavailable or longer bioavailable. Next example is a conjugate of folic acid with tamoxifen. Tamoxifen is anti-estrogen used in treatment of breast cancers, estrogen-dependent breast cancer, or another estrogen-dependent diseases. But this conjugate of folic acid and tamoxifen is even more effective in the against breast cancer, which depends on the concentration of estrogens. So in biology-driven bile acid-based drug development, many compounds are synthesized. Among them, many are very active. There are found many antimicrobial agents, many anti-cancer agents, and usually these compounds are quite antimicrobial agents are usually have quite similar, quite simple synthetic procedures. Many amines or amides, many compounds with nitrogen-containing scaffolds are very effective, and many of them are synthesized during ages or decades. There are many compounds, really many. Here are some, only a couple of examples. So in biology-driven drug development, often are present simple organic synthesis and high-throughput screening of libraries consisting of many compounds. On the other hand, syntheses are quite often in the organics, multi-step synthesis are quite often in the organic synthesis. So I believe that organic synthesis probably more like, let's say, that chemistry-driven drug development. Maybe you can say that it is more sophisticated because you have to synthesize that designed molecule, not many different compounds, but designed molecule. So in that case, compounds prepared in multi-step synthesis are usually demanding conditions. These compounds are then tested only or preferably for defect on that target molecule for which are these compounds designed, not for all other possible effects for which tests are available. Many times it's difficult to get a couple of milligrams for testing. Synthesis know that, so they are choosing tests. But from the other side, researchers never know which protein could be target, actually which protein possesses some special place or some small pocket in which some compound can bind or some scaffold can bind in that pocket and produce some useful pharmacological effect. So that was a starting point for our research, and we have got many interesting results, among which some were even unexpected. Sorry. So we wanted to find substrates or inhibitors for steroid-converting CYP enzymes among a library consisting of about 150 modified steroids. Among steroids, we had 17 substituted understand-derived substances. Most of them had a pyridyl group in position 17. Then the homolactone understand-derived compounds, diffused understand-derived compounds, the seco-estron compounds, the seco-understand compounds, then bile acid-derived substances with different number of hydroxyl function, different number of oxo function, and different number and position of double bonds. We had also some bile acids with the B or C fused tetrazole rings, and we have some bile acid-derived substances with B-homolactone moiety. So among all of these compounds, we wanted to find some ligands for CYP enzymes included in steroid converting. High throughput screening for recognizing of steroids with potential for binding to CYP17A1, CYP19A1, CYP7A1, CYP-B1, CYP21A2 was available. And in chemistry-driven drug development, androsten-derived compound should be screened for binding to CYP17A1 or CYP19A1, while bile acid-derived compounds should be screened for binding to CYP7A1 or CYP7B1. But in biology-driven drug development and in structural biology studies, all compounds, all library of compounds should be tested against all enzymes. So we tested many different compounds. Here are presented some of androsten-derived compounds, namely the seco and the homolactone androsten-derived compounds included in study. And we included also different derivatives of bile acids with different position, different number of hydroxyl groups or keto groups, or different number and position of double bonds. We also included bile acid derivatives with the free carboxyl function or their esters, butyl ester or methyl ester. And we had some acetates. So we got very interesting results. Many of compounds from the library were binding selectively to one of the tested CYP enzymes as substrates or inhibitors, but the most compounds were binding to CYP17A1 on lyase. All of these compounds which were binding as substrates. Androsten-derived compound could be supposed to bind to this enzyme, but bile acid derivatives compounds were binding even stronger with lower KDs. Bile acid derivatives compounds were binding with KDs comparable to that of natural substrates of CYP17A1, progesterone, or another. Some of bile acid derivatives were binding even with the bigger activity, bigger affinity actually, as this compound 7. So although bound as substrates, they didn't undergo reaction. They just were bound to the enzymes, and in that way they actually inhibited enzyme for its activity. So when we checked activity of this enzyme in the presence of such substrates, such ligands, actually, in some cases, enzyme wasn't absolutely ineffective. In all cases, it was less effective, but in some cases, presence of compounds, for example, 1 and 6, this compound was completely ineffective. Although bind as a substrates, they didn't go on reaction, so they just inhibited the enzyme. And this was very interesting because actually we could exclude, in chemistry-driven drug development, we could exclude bile acid derivatives for testing against CYP17A1. So in this way, we found very, very active ligands. So now I would like to say something about bile acids, bile salts in drug delivery. Namely, there are many bile acid-based drug delivery systems, and some of them are mixed micelles, some of them bilesomes, or some nanoparticles. But all of these bile acid derived drug delivery systems are based on a special kind of amphiphilicity of bile acids. Namely, these bile acids are bisphoner amphiphils, which concave, with convex beta phase, which is hydrophilic thanks to this steroid scaffold, and alpha phase is concave. It is hydrophilic, highly hydrophilic, because of present hydroxyl functions, alpha-oriented, and carboxyl function in side chain position 17. All of drug delivery systems have some advantages or disadvantages, but I think one of the most promising drug delivery system is formation of mixed micelles with drugs, with drug molecules, which enables better permeabilization of drug molecules through biological membranes. Further, it's possible to make conjugates of bile acids covalently bounded to biologically active compounds, as we saw before. So in that way, some pharmacophores can be introduced easily or more easy into the cells. There are a simple way to introduce drug into the cell. It's possible to undermine membrane stability with bile acid anions in order to deliver the drug molecule into the pathogen cells. High membrane toxicity of bile acids and toxicity in the gastrointestinal tract of patients could be a problem with the application in medicine. Still, if pathogen is outside, that problem shouldn't exist. So we oriented towards bacteria which cause skin-bound infections. And another possible direction was toxicity lowering by using 3-oxo or 3-dehydro derivatives of bile acids instead of bile acids themselves. So as a model system, we selected Acinetobacter baumannii. We selected that bacterium as a model system from several reasons. Being gram-negative bacterium, it's more challenging for treatment than gram-positive bacteria. Next, the bacterium is frequent etiological agent in wound infections. It is an important nosocomial pathogen that prolongs the length of hospital stays and increases healthcare costs. And bacteremia caused by Acinetobacter baumannii has a high mortality risk, very, very high mortality risk, actually. The most challenging, actually, was next reason, and it is that Acinetobacter baumannii has a remarkable ability to develop or acquire resistance to various antimicrobial agents, frequently being multi-drug resistant or even pan-drug resistant. So World Health Organization appointing this bacteria as a critical priority pathogen that poses a significant threat to human health. Thus, novel antimicrobial agents and strategies for combating multi-drug resistance Acinetobacter baumannii infections are urgently required. These were reasons why we wanted to test if bile salts or their 3-oxo derivatives may offer solution. For the experiments, we selected six strains of Acinetobacter baumannii bacterium, one reference strain from the ATCC collection, and 5 multi-drug resistant wound isolates previously characterized. Of particular importance was multi-drug resistant strain number 4914, because it was the highest, it possesses highest resistance. Its mix values were above the cutoff for the Acinetobacter baumannii strains for all applied antibiotics. So this strain was used further as a model along with the reference strain. For this experiment, we plan to use sodium salts of oxalic acid, deoxyxylic acid, and henodeoxyxylic acid, and their 3-oxo or 3-dihydro analogs. In the experiment, we included ciprofloxacin, gentamicin, and polymyxin B, like antibiotics, with different effect, actually different resistance for different cause resistance of this bacterium. So when we tested antimicrobial effects of single agents, bile acid salts and derivatives, and antibiotics, we got very interesting results. Namely, sodium salts of deoxyxylic acid was very effective on reference strains, but also on multi-drug resistant strain. Other bile salts were quite effective, actually moderate effective, except sodium salts of oxalic acid and its 3-dihydro derivative. When we was checking activity of antibiotics, the most effective was polymyxin B, while ciprofloxacin was moderate effective, but gentamicin wasn't absolutely ineffective. So this bacterium expressed showed high resistance to against this compound, this antibiotics. Next, we selected, we excluded oxalic acid salts, and we tested combined effects of bile acids. We tested combined effects of bile acid salts and each of antibiotics. So this was very interesting because ciprofloxacin showed a synergistic effect with sodium salt of deoxyxylic acid against reference strain, but not for multi-drug resistant strain. On the other hand, all salts combined with polymyxin B were not effective against reference strain, but were very effective. Actually, we had we got a synergistic effect in multi-drug resistant isolate strain. Gentamicin wasn't effective at all when used alone, but in combination with bile acid salts was very, very effective, and the best synergistic effect actually we found in these combinations of gentamicin and all salts treated in both strains, in multi-drug resistant strain as well. So the best effect definitely was for gentamicin, and after that with ciprofloxacin. And less effective was this synergy with polymyxin B. We suppose that it is because of different way of action, mechanism of action of these antibiotics, because gentamicin and ciprofloxacin have to enter cells and then can be effective. They have to get into the cell, but while polymyxin B is acting on the cell membrane. So we suppose that bile acid salts or derivatives are effective in that way that they help, they undermine membrane stability of the bacterium, and they help these antibiotics, gentamicin and ciprofloxacin, to get into the cells, into pathogen cells, to check if they make co-aggregates, or actually, sorry, to test, to confirm detected synergistic interactions between conventional and alternative antimicrobial agents. We did time kill assay. So we checked actually what is happening during different periods, and we confirmed detected synergistic interactions between some of these bile acid derivatives, bile acid salts, and different antibiotics. To check if bile acids and antibiotics are making mixed micelles or co-aggregates, we performed the NMR experiment. So there is an NMR experiment in which we checked mixed micelle or co-aggregates formation in different with combination of two of antibiotics and bile acid salts, but where bile acids were in different concentrations. So log D values are these which are showing that compounds make mixed micelles or not. So we actually found that mixed micelles are formed only above critical micellar concentration of bile acids, while in synergism experiments, concentrations of bile salts were quite lower than critical micellar concentrations. So in these synergistic experiments, bile acids, bile salts probably didn't form co-aggregates, but enhanced ciprofloxacin penetration into the cells via membrane destabilization. We performed this experiment only with ciprofloxacin because it is the smallest molecule of all used antibiotics. So it was more was let's say most appropriate to make mixed micelles. So co-aggregates were organizing above critical micellar concentrations. So we in that with this experiment actually didn't show that micelles are not organized, but we suppose that they are not organized in this lower concentration. So taking into account the strain's origin, it's obvious that the obtained synergistic interactions should be further examined in vivo experiments and in clinical trials, particularly with gentamicin, where the highest resistance is achieved actually. And the synergistic concentrations can be easily obtained topically for treatment of infected wounds, so should be further examined using corresponding in vivo models. Here, these results were achieved in the cooperation of colleagues from research group for the synthesis and testing of biological activity of steroid compounds, Department of Chemistry, Biochemistry, Environmental Protection, Faculty of Sciences, University of Novi Sad, which is existing and synthesizing steroid molecules more than 50 years. So our research group, my research groups, did these experiments with colleagues from Institute of Bioorganic Chemistry, National Academy of Sciences of Belarus, and Department of Biology and Ecology, Faculty of Sciences, Novi Sad. So thank you for your kind attention. Дякуем вам за увагу. Благодарю вам за внимание.
Okay, thank you, Susanna, for nice presentation showing how bile acid may be the precursor of the drugs and drug delivery system. We have a chance for one question. No one. Okay, thank you again. I am very impressed by your data that bile acids, 27 steroids might be the ligand for the CYP17, which is a normally oxidized C21 steroids. It's very interesting and wonderful results. These steroids interact with the cytochrome P450 with a high affinity equivalent with the normally used drugs. So from my opinion, it's very important result. Thank you.